Thank you for the invitation. And before we begin, I'd like to thank the Rappaport Foundation for their generous support. So, the most recent estimate are that 1 in 59 children born today will go on to be diagnosed with closer? All right. Will go on to be diagnosed with autism spectrum disorder. Now, this number actually represents a dramatic rise in prevalence in recent decades. And whether or not this rise in prevalence is due to things like more encompassing definitions of what is autism, an increasing awareness of the disorder. There have been a number of successful campaigns that have done, really enormous benefit by bringing the disorder to the forefront, or whether it is due to some form of genuine increase in disease prevalence. So whether or not, all other things considered, the disorder is becoming more common, a very controversial and difficult thing to actually say. I think ultimately what is most important is that what this number represents is really a substantial need for both really just a better understanding and for new options for treatment. So classically, autism has really been defined by a set of behaviors, things like problems, social impairments, problems with communication, and what are known as these repetitive or stereotyped behaviors. But one of the things that we're learning is that many of the common co-occurring medical conditions, so these are things like seizures or epilepsy, things like altered sleep patterns, or things like gastrointestinal issues, these may actually arise from the same underlying cause. And if we fully want to understand this condition, we need to better look at these, at this broader picture. And additionally, the other thing to know is that, whenever you give something a name of spectrum, it means that it is incredibly diverse and it looks very different person to person. And part of the reason we suspect this is because we think that there might be actually multiple kind of causes and multiple factors that kind of lead to autism. Now, broadly, you can kind of divide these into either genetic, those that are kind of heritable, or the remaining portion, which we're going to call environmental. Now, there's been, phenomenal progress and an intense area of interest in the genetic forms of autism. And we've learned a lot in recent years, including that there is, in most cases, it's an incredibly complex picture. It's typically not just individual genes, but hundreds of genes all kind of interacting. But we've also learned that you can estimate that this genetic proportion probably only accounts for somewhere between 50 to 80%. A large portion, but not all of it. And we're particularly interested in that other causes, the environmental. We're interested in one in particular, the potential role for inflammation, or the involvement of the immune system. So now, evidence for this is really coming from studies that have looked at the brains from those with autism. And now, in some of those, you'll find that in the brain, you can actually see changes in some of those immune pathways. Additionally, if you look in the brain, you can find a very special cell type. It's a cell type known as a microglia. And in a healthy, normal brain, this is the only kind of immune cell type you'll find. Now, in some of those with autism, we'll find that not only are there more of these microglia, but they actually physically look different. And to some extent, this all makes sense in the bigger picture. Because what we've learned in recent years is that we were wrong. We used to kind of believe that here's your nervous system, here is the immune system, and they were kind of like neighbors in Boston. Live right next to each other, but they avoid talking to each other at all possible cost. We now know that is not true. We now know there's this elaborate crosstalk between the immune and the nervous system. And the reason why that's important is because now you can imagine the situation where you have the growing, developing brain, something that is just so unfathomably complex. You have to imagine there are billions and billions of connections that all have to form in the right way, at the right time, and at the right spot. And you kind of throw a wrench at that with something like immune activation, with something like an infection, like influenza. And you could see how this might be able to essentially change the course of that developing nervous system and lead it towards a different destination. And this may ultimately result in something like autism spectrum disorder. And in fact, you can go to the clinical data and you can look at these massive data sets. And these are epidemiology studies, and you can find that for those who had an infection, so something like influenza or a bacterial infection during pregnancy, the child of that pregnancy was at an increased risk for developing autism. Now, important disclosure, increased risk is not a guarantee. In fact, most of those children don't develop autism for reasons we still don't completely understand. But what we do is we attempt to model this in the laboratory. And to do that, we use mice because mice allow us to look at a really kind of controlled manner because we can choose exactly when the mice get pregnant, and then we can use various agents that kind of mimic infection and target particular immune pathways. And we can look at how this affects the developing nervous system. And sure enough, we find that certain types of immune activation can lead to altered phenotypes in these mice. So what we do is we do these kind of behavioral assays where you have these mice interacting with a partner mouse and kind of just get an idea how much is interacting. And yes, these have been kind of the state of the field. And they do provide information. But you can imagine it's quite difficult to guess, really infer exactly what a mouse is thinking and exactly what the intent is. So to really kind of move beyond that, we've started using these little guys. And it doesn't look like much, and I bet some of you in the back probably can't even see it, but within this little thing is an enormous amount of miniaturized technology. We can implant these, you essentially put them in the body cavity and you can suture it up, and we can get an enormous wealth of data. So we can hook this up and we'll get brain activity from EEG, we'll get muscle activity, we get temperature, we get how much the mouse is moving around, and we can use all this to look at the sleep-wake states of the animal. And sure enough, when we do this to these mice, we can see there are some signs that do look somewhat similar to autism. We see the seizure-like brain activity. We see changes in sleep, and we see changes in how the animal is moving around. And now that we have these measures kind of established, we're really kind of an opportune time to test if various kinds of interventions are able to change any of these measures. So for instance, one study we're doing is, you remember those microglia, those immune cells in the brain? Well, it turns out there's a drug under development that has the ability to essentially eliminate those microglia, and then you take them off the drug and they regrow. So, and when you kind of deplete and regrow, you can kind of resolve some kind of ongoing inflammatory processes. You can kind of think of it as like restarting a computer. You know, don't entirely know the full process of how it works, but it's going to be the first thing your IT person asks you to do anyway. So I think what we ultimately hope is that by studying the system, not only can we better understand these kind of potential mechanisms, hopefully for maybe a small subset of patients will even lead to entirely new strategies for treatment. So thank you.